So good morning, and so having heard about the importance of sleep, the functional importance of sleep, and so subsequently now we'll try to understand the intricacies of the neural substrates of sleep, and this will be followed by understanding the basics of the neurophysiology of sleep as well. So we know that sleep is a universal phenomena, across the animal kingdom, and Madam has elaborated upon that. And of course, the sleep is the resultant of the interaction of many micro and macro, the networks across the brain and also in the periphery. So genes plays an important role, connections plays an important role, circadian rhythm plays an important role, and our behavior plays an important role. So all these things has to be integrated to bring about a proper sleep. So how do we define a sleep? So the mammalian sleep is defined as it is a reversible behavioral state of perceptual disengagement from and responsiveness to the environment, which has a 4 criteria. It has to have a reduced motor activity. It has a reduced response to any stimulation. There should be a stereotypic postures, especially in the humans. It has to be a supine posture, and it has to be easily reversible. So these 4 things plays an important role to define the behavioral definition of the sleep. So as we know that, when it comes to the human sleep, so it has been broadly divided into NREM sleep and REM sleep. And in turn, the NREM sleep is divided into the 3 different stages, N1, N2, and N3. So N1 and N2, they're called a lighter sleep stage. N3, we can call it as a deep sleep stage. And we have a REM sleep stages. So this is a very typical sleep architecture or the hypnogram that is expected from any healthy adult human being whenever we record the polysomnographic data for the whole night. Now this whole sleep is controlled by 2 important processes. One is the homeostatic process, the another is the circadian process. The homeostatic process is that particular process where, as in the daytime, when we go on, we engage in our work, the sleep pressure, it goes on build up. But so when this pressure of the sleep goes on, it builds up, then that will increase the pressure of the sleep, and this particular pressure is what that drives for us to get into sleep. So if you can see here that when we get up in the morning and when we start our day activity, the sleep pressure it goes on increasing, and as the sleep pressure goes on increasing, when we get into the subsequent sleep, it is the amount of slow wave sleep or the deep sleep which will be proportional to the amount of sleep pressure that has been accumulated in our day activity. Suppose if we have for any other circumstances, if we have sleep restricted or the sleep deprived, and if there is an additional sleep pressure where we have missed the sleep, right, and then subsequently also, right, there will be more slow wave sleep. So our slow wave sleep or the slow wave activity or the deep sleep is proportionate to the amount of sleep pressure that has been accumulated in our day activity. So therefore, this dissipation of the sleep pressure during sleep, which is proportional to the deep sleep or the slow wave activity, is very, very important for the many physiological functions that is which is associated with deep sleep state or that of a slow wave activity. So this whole process is called as homeostatic regulation of sleep, which is of which the sleep pressure is very, very important. The other thing is about the circadian rhythm, where we can see that with our 24 plus hours, which is dependent on our exposure to the sleep, dark, light dark cycle. Okay, we have the circadian rhythm, and the way the Zeitgeber, especially the light, so which comes to the suprachiasmatic nucleus, in turn the network from suprachiasmatic nucleus, both at the central and at the peripheral, how it modulates the various, the proteins, protein expressions, hormones. So all those things also plays a very important role in regulating sleep. So these two processes, homeostatic and the circadian, plays a very important role to have a very adequate sleep for us. So in this neural subset of sleep, I'll just focus on the homeostatic regulation of sleep. So this slide gives you the overall say intricacies how the circadian and the homeostatic pressure or homeostatic regulation, so they are very highly linked with each other, right, to bring about not only about the sleep, but also it influences the autonomic activity, it influences the functions of the various peripheral physiological, these functions. It helps in the expression of various proteins for the maintenance of the cellular health, and also it is involved in various, including the food, like the food intake, exercise, all these things. So we have both the circadian as well as the homeostatic pressure that plays a very important role. When it comes to we request all of you to please mute yourself. So before trying to understand the neural subset of sleep, so I'll give you in brief about the network that will be responsible for inducing the sleep. So all of you know of us, we know that the ascending reticular activating system that play a very important role in maintaining of our wake, and which is made up of two important the neurotransmitter system, the cholinergic system with and also with the monoaminergic system. Now the cholinergic system over here, it activates the cortical areas and it, via the thalamus, and the monoaminergic system, right, it activates the cortical areas via hypothalamus. Now both these, they activate the forebrain and it helps into maintenance of the wake. Now, so with these wake promoting neurons, that what we are seeing here, the just the in detail of this I'll take in the next session, but for us to understand something on the activity of the sleep substrate, we need to have the basic understanding of this. Now subsequently, here also we have one of the neuronal group, that is a ventrolateral preoptic area, which is a sleep promoting neurons. So both wake promoting and the sleep promoting neurons, they are very adjacent to each other. And these ascending reticular activating system by cholinergic and monoaminergic, they activate the forebrain, which helps us to maintain the wake state. So then when it comes to the sleep promoting neuron, that is ventrolateral preoptic area, so that will send the inhibitory input to all the wake promoting neurons, and ventrolateral preoptic area is more GABAergic, So because it is more GABAergic, so whenever VLPO is very active, it inhibits the wake promoting neurons so that sleep is maintained. So therefore, this VLPO, that is ventrolateral preoptic area, which will be very active during sleep, is considered to be, it is involved in the maintenance and execution of sleep rather than driving for the sleep. So here we have two important aspects. One, the factors which drives our system to get into the sleep. The other is the factors which helps us to maintain the sleep. Now, for us to drive into the sleep is what the homeostatic mechanism, which plays a very important role, which I've told you. And for the maintenance of the sleep is all this sleep related circuitry plays a very important role. So as far as the neural substrate is concerned, now we'll try to concentrate only on those factors which helps us to drive for the sleep. So when it comes to the sleep regulating substances, so it was the Japanese scientist who discovered that there are some substances which can induce the sleep. So they homogenize the brain extracts from the sleep deprived dogs, and when this was injected to the normal dog, the normal dogs spend more time during sleep. Then so they inferred that, and they first demonstrated that there should be some substances or endogenous substances, so which will get accumulated in the system whenever there is a sleep deprivation, and when this is given to a normal animal, and that will induce the sleep. So there should be some substances in the system. Now, so these sleep regulatory, so there are the criteria where we can say, okay, this is a substance which is related to sleep. So what are those criteria to say that, okay, these are sleep regulatory substances? Number one, it should induce and maintain the physiological sleep. Number two, its receptor should be present. Number three, so whenever there is an inactivation of sleep regulatory substances or its receptor, so at least temporarily should reduce the spontaneous sleep, and also possibly it should attenuate the sleep response to a somnogenic stimuli. So one, it should induce and maintain the sleep, and there should be a specific receptor for the particular, the substance should be there, and inactivation of these things, okay, it should attenuate the sleep at least temporarily. And most important is the brain levels of these substances should vary according to sleep-wake cycle. So whenever we are in the wake behavior, these sleep regulatory substances should come down, and whenever we are in the sleep, or these sleep regulatory substances, it should increase. And not only that, there should be a variation in the sleep-wake cycle. These substances should act on the sleep circuit also. So which I briefly told in the past few two slides. And these substances should also be altered in the pathological states that are associated with enhanced sleepiness. Even it can be like a physiological state like a sleep deprivation or the sleep restriction. So still that we have extended sleep restriction, these particular substances should enhance whenever the sleep pressure is increasing. And also other important is, though these sleep regulatory substances could be associated with various other physiological functions, but the somnogenic function, right, should be exclusively, it should be a separate. So these are the criteria for any given substances, endogenous substances, which we can label it as sleep regulatory substance. So many sleep regulatory substances have been listed, which act specifically with NREM sleep and REM sleep. So in NREM sleep, regulatory substances are like adenosine, nitric oxide, all the cytokines like prostaglandins, tumor necrosis factor alpha, interleukins, hormones like GHRH. And REM sleep is associated with prolactin and VIP, and the wake regulating like it will be like orexin, ghrelin, ACTH, CRH, okay, all these things. So of all these things, adenosine is one of the main important fulcrum, right, which will try to regulate the sleep, or which will try to initiate the sleep, and which plays a very important role in the homeostatic regulation of sleep. So we'll try to understand something more on the adenosine in the subsequent slides. So having known the list of all the sleep regulatory substances, so as I said that these, one of the criteria for the sleep regulating substances is that it has to act for the sleep wake regulatory circuits. Now, so the adenosine plays a very important role, and its main the area of action is in the basal forebrain, and the cytokines like tumor necrosis factor and interleukins 1, it acts both in the hypothalamic preoptic and the locus coeruleus, where locus coeruleus plays a very important role in both REM sleep and also in the wake, and GHRH plays an important role in the hypothalamic preoptic area. And the Raphe nucleus that plays also very important role in GHRH and interleukins 1. So the adenosine plays a very important role at, and the site of action of adenosine, right, will be the basal forebrain. So of all the sleep regulatory substances, adenosine, prostaglandin D2, and cytokines, right, they act at the molecular basis on the sleep homeostat, and it interacts with the sleep regulatory network. And so as another criteria is that among all the sleep regulatory substances, that we can see that it varies according to sleep wake cycle, where it increases during sleep, and as the sleep pressure dissipates, the level of these sleep regulatory substances, it also reduces. Coming to adenosine. So adenosine, as we know now, it acts, the predominant area of action of adenosine is on the basal forebrain, and it has various receptors that it acts, but out of which A1 and A2 plays a very important role as far as the sleep is concerned. And but important is adenosine, it regulates sleep by both inhibitory and also with the excitatory activity. So inhibitory activity is driven by A1 receptors, and excitatory activity is driven by the A2 receptors. And A1 receptors, right, it is widely distributed both at the presynaptic and at the postsynaptic areas. And the action of A1 receptor is by it inhibits the wake promoting activities of the basal forebrain, and it promotes the activity of VLPO. So now by this time we know that VLPO is a sleep promoting, and basal forebrain is important. Inhibition of the basal forebrain is important for the activity of the basal forebrain is important for the wake. So therefore A1 by its inhibitor activity, it inhibits the wake promoting activity of the basal forebrain, and it promotes the VLPO activity so that the sleep is initiated. Then A2, it acts through the excitatory mechanism, and it acts on the striatum nucleus accumbens, and also about the basal ganglia and so many other areas. And we'll try to understand how these both A1 and A2 brings about the sleep. And of course, like any other cytokines, adenosine also show a very diurnal, these variations. The source of adenosine, so is with the interaction between neuron and glia. So where the level of adenosine, the concentration of adenosine that gets accumulated is proportionate to the demand for the oxygen. That is, when the supply and demand ratio for the oxygen, it goes on decreasing, right, the adenosine level starts increasing. So it means that whenever these the activity or the neuronal activity or the interaction between the neuron and glia, right, is more than that of the optimal, right, and because of the metabolic activity, adenosine, it goes on accumulating. So with the activity at the synaptic level, the ATP that is generated, right, it can be converted into ADP, AMP, and AMP via acting on the enzyme CD73 on the neuron, right, it is converted into adenosine. Similarly, intracellularly also, so there will be adenosine, and then this adenosine, so the exchange of adenosine both from the intracellular and the intracellular, so it happens to the enterocyte, these channels. So the concentration of adenosine both intracellular and extracellular has to be, it has to be maintained. So similarly, it also takes place in the glial cells also, where the activity of the glial cells with that of the where the adenosine increases with adenosine kinase, it is converted into ANP and then again to ATP, and ATP is driven back. Again, this ATP is converted to ANP, and at the end, the ANP again is converted to adenosine. So this concentration of the extracellular adenosine is directly proportional to the degree of the interaction that takes place between the neuronal and glial interaction. And as the oxygen demand, it decreases, this adenosine level increases. So once the adenosine level, it starts increasing in the extracellular or the extracellular concentration of adenosine, it goes on increasing, then the adenosine plays an important role in bringing down the activity of the neurons, and it drives for the sleep. So again, this explains about the various pathway in which this adenosine gets accumulated, and how and it acts on both A1 and this A2 receptors. So if you look at even this particular graph, so it tells that whenever there is a sleep deprivation, so in all these areas which are associated with the sleep-wake circuits, the concentration of adenosine, it will increase. But however, the highest concentration, right, you can see in the basal forebrain. And whenever there is a recovery sleep, so right, as the sleep pressure dissipates, so we can see that adenosine, it goes on reducing in all the areas, and also it reduces in the basal forebrain also. So as it was told that when the receptors of the adenosine has its expression in both presynaptic and the postsynaptic, so it means that adenosine induces sleep by both the presynaptic activity and also with that of the postsynaptic activity. So how does the adenosine helps to bring about the slow wave activity or the sleep? By the presynaptic activity. Now, once the adenosine is released because of the activity of the neuron and glia, now the adenosine, okay, it acts on its presynaptic, the receptor, right, and inhibits the release of the glutamate. So once there is an inhibition of the release of the glutamate, it increases the expression of the potassium channels and increases the conductance of the potassium channels. So once there is an increase in the conduction of the potassium channels, this presynaptic terminal get into the hyperpolarized state, and there will be a functional differentiation of all the presynaptic terminals with that of the postsynapse. And where this presynaptic differentiation, functional differentiation takes place, it takes place at the thalamic level, when it plays at the cortical level. So at the local level of both at the cortex and at the thalamus and the thalamocortical, these areas. So whenever because of the presynaptic inhibition of the release of the glutamate by the adenosine, and the increase in the expression and the conduction of the potassium channels, so all the there will be the functional differentiation between thalamus and the cortical neurons, and in turn it induces the local slower activity. So this is how the adenosine plays an important role in functional differentiation by its presynaptic activity. But when it comes to the postsynaptic activity, so it will be a cascade of events, so happens at the postsynaptic level. Now, even when the adenosine gets accumulated, okay, the interaction between the neuron and glia, and also the ATP is also generated. Now, the accumulation of ATP, right, at the synapse, it acts on the glial purinergic receptors, and in turn, because of the action of the purinergic receptors, it will release all the cytokines, especially tumor necrosis factor alpha and the interleukin beta, and these two in turn, right, acts on the postsynaptic the postsynaptic terminal, where the inactive nuclear factor will be converted into active, and this active nucleus factor is what it expresses more and more adenosine receptors at the postsynaptic junction. So that is to say that, and when there is a more postsynaptic expression at the of A1 receptor in the postsynaptic terminal, whatever the adenosine that gets accumulated in the extracellular, it acts on the A1, and in turn, the when adenosine binds to A1, right, again it activates the potassium conduction even at the postsynaptic terminal. So adenosine, both from the presynaptic per se by inhibiting the glutamate release and by increasing the potassium conductance, and at the postsynaptic, right, where by ATP induce the cascade of events by the release of these cytokines, right, and by increasing the A1 receptors, adenosine A1 receptor at the postsynaptic level, so it will also reduce the activity at the postsynaptic level. So this is how the postsynaptic scaling or the downscaling of the postsynaptic activity in all these the wake promoting and the inhibition of the wake promoting circuitry, it depends on the amount of ATP, that is triggered by the presynaptic activity. So this is how the whole metabolomic things is related to the initiation of sleep and by or the inhibition of the wake promoting activity with the circuit that is related to that of the wake, where both presynaptic and postsynaptic way in which the adenosine plays a very important role. And the other also is that the interaction between the astrocyte and the neuronal glia, right? It depends on, it depends on the amount of calcium that gets accumulated because of the activity. Now, as the intracellular calcium, okay, it increases because of the which is directly proportional to the intensity of the activity, that at the cellular level. Now, my sleep process, that is process S is nothing but the homeostatic process. So it goes on build up. So the interaction also between neuronal and the glial interaction, right? It also depends on the amount of calcium that gets accumulated in the by the intracellular calcium, the accumulation, which in turn which drives the further pathway with diacylglycerol, inosyl triphosphate, right? All those things, and which will drive the glio, the transmission. So this is how in brief where the adenosine acts through A1 receptors. When it comes to the A2 receptor, so A2 receptor is more expressed right in the nucleus say this accumbens, and A2 receptor is more active or is more excitatory, in nature, where adenosine will excites the has an excitability action on A2 receptor. And nucleus accumbens, we should know it is more GABAergic in nature. So whenever there is a activity of nucleus accumbens, right, in turn this will inhibit all the wake promoting the neuronal group. So we have this ventral pallidum, lateral hypothalamus, parabrachial nucleus, ventral tegmental area. So all these things in turn, all which is involved in the wake promoting activity. Especially you can see that this lateral hypothalamus in turn is connected to the sleep circuitry, which I showed you earlier, where we have VLPO, locus coeruleus and TMN nucleus and all, right? So the activity of adenosine on the nucleus accumbens by exciting by A2 receptor, which in turn inhibits all this the wake promoting neuronal group. Now, by inhibiting this wake promoting neuronal group, right, that the sleep is so induced. So this particular model where nucleus accumbens, right, which predicts that adenosine that acting on by A2A receptors, where and the activity of adenosine is much more powerful, right, than the inhibitory action of ETA on to this of the D2 receptors on the nucleus accumbens. So thus the adenosine by A1 and A2 receptor, by both the presynaptic and postsynaptic through the A1 receptors, and directly on A2 receptors with the nucleus accumbens. So it inhibits all the wake related circuitry and by initiating that of the sleep. So this will give the full summary. So whenever that because of the activity, when adenosine is generated, right, it acts on the A1 receptor directly, and also the purinergic receptor, so which will induce all the cytokines which are related to generation of NREM sleep. Further, the same purinergic receptors, right, when it acts on the P2 receptors, right, it releases the prolactin, the hormone, which is responsible for that of a REM sleep. So with all this cascade, one of the common factor is that of a adenosine. Now, having known in brief about adenosine, we'll try to understand something also on the prostaglandin, which also plays a very important role in inducing the sleep. Now, in the normal physiological doses, prostaglandin D2 can also induce both NREM sleep and the REM sleep. So whenever the normal physiological dose is administered and the sleep, and when it initiates the sleep, it is very difficult to distinguish between the normal sleep and also sleep that is induced because of the prostaglandin D2. So that is to say that even in our day-to-day activity, for all the wear and tear mechanism, right, when this particular cytokine or this prostaglandin, gets accumulated, it can induce the normal, the physiological sleep. And also, so studies have very clearly shown even this prostaglandin D2 also varies along with the sleep wake cycle, and also it is involved in the circadian regulation of sleep as well. So how does this prostaglandin D2 it initiates the sleep? So in all these meninges, leptomeninges, choroid plexus, at the corpus callosum, right? So we have a very important enzyme, right? Prostaglandin D synthase enzyme, where the prostaglandin D2 is synthesized. Now, this prostaglandin D2 plays a very, very important role in the induction of, the sleep. Now when prostaglandin this D2 is synthesized in these choroid plexus, right? It through the arachnoid and tubercular cells, right? It enters to the CSF, and in the CSF, as the concentration of the prostaglandin increases to that of an optimal, the level, so it acts by the two mechanisms. Number one is that it activates the sleep promoting neuron, that is VLPO, and it inhibits the wake promoting, that is histinergic network. So prostaglandin D2 plays another very important role by inducing the sleep, and also prostaglandin D2, it upregulates the sleep inducing neurons, right, in VLPO, and it downregulates the wake promoting neurons in tuberomammillary nucleus. So again, so this slides will tell you about how the prostaglandin D2 and the adenosine, they gets interacted with each other. So as I mentioned earlier, in all this cascade of the cytokine pathway, see, the one final common in which the sleep is induced is always through the adenosine. So we have prostaglandin D synthase. Prostaglandin D2 is increased in the CSF. So as when the prostaglandin D2 gets accumulated, right, it acts on the prostaglandin 1 receptor, and this in turn increases the adenosine, and further down the cascade, as I mentioned earlier, where adenosine can act on A2A receptor by activating and activating VLPO, that is sleep inducing, and inhibiting the wake promoting. So again, with this, right, this adenosine plays a very, very important role. So apart from prostaglandin D2, so we have many other cytokines, so which will follow their own specific pathway, but the common pathway is always that adenosine acting on both A1 and A2 receptors. Then, so, and both, the other thing is also that these pro-somnogenic cytokines, so it acts by the inhibiting the and also inhibition of the arousal system, and also directly activating the inhibitory system. Inhibitory system here means the sleep promoting system. Arousal system is the wake system. So now all these pro-somnogenic cytokines, including nitric oxide, prostaglandin D2, at the end of the day, it's adenosine. So where adenosine, by inhibiting the arousal system by A1 receptor, and directly sometimes these cytokines acts on activating the inhibitory system, right? It can induce the sleep. So this will give you the whole network, that how the even at the molecular level, right, the accumulation of the metabolites can induce sleep with the activity of where it can generate out of these cytokines, right, where it can in turn induce the nitric oxide, right, in turn the adenosine, right, and again it can act at the various levels. So it can be at the neuronal assembly levels, right, it can inhibit the excitatory neurons, activate the right, that of inhibitory neurons. So this whole cascade, which can start from at the genetic level for the expression of the receptors, in turn, right, it brings about the various changes in the synaptic transmission, and because of the sleep as such, there will be an increase in the intercellular or interstitial space, where it can even increase the blood flow, or so that these waste products are drained out, as we heard with madam's lecture earlier, that we know that during sleep, right, our interstitial space, okay, is increased, and where which will facilitate the clearance of all these metabolites. So even this clearance of these metabolites, and with the increasing of the interstitial space, right, is what it is facilitated by the nitric oxide synthase or here, and which is also in turn facilitated by the accumulation of all these cytokines. So so this is to say that the sleep pressure or the homeostatic regulation mechanism of sleep, so which plays a very important role for the initiating of the sleep. So, and as our day-to-day activity increases, depending on the effectiveness or the efficiency of our neuronal and glial interaction and the demand, so there will be accumulation of ATP, and this ATP in turn brings down the cascade of events, both acting on the glial cells and bringing about the cytokines, and in turn the adenosine acting on both pre and postsynaptic level, and where it activates the sleep promoting and inhibit the wake promoting the activity, and in turn the cytokines can also bring about increasing nitric oxide synthase or the nitric oxide, right, which will help in increasing the interstitial space, and which helps in to the effective clearance of all the metabolites that have been accumulated. So this is in just which wanted to share with you about the neural substrate of sleep, especially concentrating on how the homeostatic regulation of sleep as far as NREM sleep is concerned. So thank you very much. So I'd like to take any questions. Is there any role of BDNF? Now, we know that the role of BDNF is, of course, it plays a very important role in the neural plasticity and also with the bringing about cognition and all. So definitely, whenever there is a sleep deprivation, because of the sleep deprivation, the BDNF accumulation or the BDF concentration has the it will definitely it should reduce. But is there any role of BDNF as far as the initiation of the sleep is concerned? I'm not very clear about that.